Plasma membranes dictate what enters and exits the cell. They are selectively permeable. And this is important because without selective permeability, the cell would take in anything and leak out everything. It is the very thing holding the cell together both physically and chemically. And this separation of the extracellular fluid and the cytosol on the inside allows cells to carry out chemical reactions that could not have been done if bathed in the same fluids as the extracellular fluid. The good news is that selective permeability costs the cell nothing, provided that materials move from an area of higher concentration to lower concentration. The physical space between areas of different concentrations of a given substance is called a concentration gradient. And sometimes materials need to move against the concentration gradient, and this requires ATP. Not all substances pass through the cell membrane equally. Smaller, nonpolar molecules like oxygen and carbon dioxide can pass through the membrane with relative ease, but larger and/or polar substances such as glucose and many proteins require special channel and carrier proteins to allow them passage into the cell. Ions, or atoms with charges, also present a problem as the phospholipid bilayer repulses charged things, even if the molecules are small enough to fit through the phospholipids. And like larger and polar molecules, these require special channel proteins to help them pass through to the inside of the cell.
In this video, we will focus on passive transport. That is, when substances move from an area of high concentration to low concentration and bears no energy expenditure on the cell. And that's because this is a naturally occurring phenomenon that exists regardless if cells are present or absent. Diffusion is the movement of molecules from an area of high concentration to an area of lower concentration to achieve equilibrium of concentration. You don't need living cells to perform this process, as you experience diffusion all the time. I'll use this demonstration with food coloring and water. The food coloring, which is highly concentrated, goes through the container of water, which initially has no food coloring. It begins by going downward and outward, and as it congregates at the bottom, it then moves upwards. This pattern will continue until the entire container is perceived by us to be the same color. This is equilibrium, or the even distribution of a substance in a given area. Now keep in mind that molecules do not stop moving. They are just distributed more evenly. You see this with the sense of smell as odors travel across an area. In fact, it's probably true you've been in a situation like this one.
Oh my God!
Put the window down!
I don't know what you guys are talking about. I think it's fine. Oh! Oh my God! Oh! Open the window! Please, now! For God's sakes, open the window!
Okay. Oh! Now let's use science to explain how diffusion of those fart molecules went around the car. The driver occupies one area of the car. The highly concentrated smell then dissipates to the passenger to their right and eventually to those in the back. Likely the person farthest from them will smell it last. True equilibrium does not happen because of the ventilation in cars and the fact that our noses tend to block out smells after a while. In cells, diffusion occurs when small, non-charged and non-polar molecules like carbon dioxide and oxygen build up on one side of the membrane and simply pass to the other side until equilibrium is met. Since you are a living thing, the concentration gradient changes consistently since you are constantly bringing in oxygen and exhaling carbon dioxide. And several things can affect rates of diffusion. First is the difference in concentration gradient. If one side of the cell has a huge concentration of a substance compared to the other, rates of diffusion will increase. Likewise, if both sides of the cell have fairly equal amounts of substance, the rate of diffusion decreases. Larger molecules diffuse much more slowly than smaller molecules, as larger molecules must move past the molecular structure of phospholipids. Kind of like a larger person having a hard time trying to get through a crowd versus a smaller person. In addition, Warmer temperatures increase rates of diffusion, and as solvent density increases, rate of diffusion starts to decrease. So what do you do with larger charge and/or polar molecules? Remember the proteins from earlier? Some of them serve as either channel or carrier proteins, which help to transport these sorts of molecules across cell membranes. This works the same as simple diffusion, as the concentration gradient is still high to low and requires no ATP on this part of the cell. Channel proteins are selective of the molecules that are appropriate size and charge. And one special type of channel protein is called an aquaporin, which helps to move water across plasma membranes. Carrier proteins are membrane proteins that physically bond to specific substances, changing the shape and moving them across. Here, the wrong molecule comes in contact with the carrier protein and no reaction occurs. But when the appropriate one bonds, the carrier protein brings the molecules in, provided that the concentration gradient is going from high to low. Now, some carrier proteins act as pumps, which require ATP, and this is a form of active transport, which I'll talk about in my next video. Osmosis is the diffusion of water across a selectively permeable membrane. Technically speaking, diffusion occurs with or without membranes, and like diffusion, osmosis is a form of passive transport and requires no ATP to operate. Water moves from regions of low solute concentration to regions of high solute concentration. In other words, water always moves towards the greater solute concentration, whether it's on the inside of the cell or the outside. This movement dilutes the higher concentration, thus equalizing the concentration of the solvent on both sides of the bilayer. Next, I want to bring up a concept called tonicity, which describes how an extracellular solution can change a cell's volume by affecting osmosis. Osmolarity describes the solution's total solute concentration. If you remember the rule that water always follows the greater solute concentration, you can predict how water moves in and out of cells. There are three general types of osmolarity that cells are exposed to. Let's assume that each cell's cytoplasm is 99.1% water and 0.9% solutes. And these solutes include things like electrolytes, proteins, waste, organic materials of all sorts. In isotonic solutions, a cell's extracellular fluid solute concentration matches that of the cytosol within the cell. This causes no net change in volume to the cell as water enters and exits the cell at the same rate. Here are human blood cells in saline. Note the circular shape. In hypertonic conditions, cells are exposed to extracellular fluid that has a higher solute concentration than the cytosol. And since equilibrium must occur, the cell loses water to the surrounding environment and shrivels as a result. And this can lead to deadly dehydration over a long period of time and a loss of diffusion of solutes across a membrane. Now look at these red blood cells exposed to salt water and note how shriveled they appear. You can also see this in plants as the cell membranes physically detach from the cell wall and the overall plant will appear wilted. Finally, hypotonic conditions occur when cells are exposed to an extracellular fluid that has a lower solute concentration than the cytosol. Since equilibrium must occur, the cell gains more water from the surrounding environment and swells. Of course, if this happens too much, the cell membrane can burst open. Because of tonicity and osmolarity, organisms have made several adaptations that allow them to survive these conditions. Remember that wilted plant from earlier? We know it's due to a lack of water, but the wilting is caused by the vacuoles shrinking in hypertonic conditions. Plants do best in hypotonic conditions because it's difficult to damage the plasma membrane since the cell wall is there to support it. This also allows the plant to stand more firm, creating turgor, which occurs when a plant's cell membrane pushes up against the cell wall, providing more reinforcement. Fish are really interesting. Saltwater fish have to deal with hypertonic conditions. Therefore, they produce a urine that is highly concentrated of solutes and less water to help retain the water and void those excess solutes. Freshwater fish have the opposite issue. They live in hypotonic conditions and must have urine mostly made of water, and the kidneys help retain more solutes. Although much of the selective permeability of cells is done without using energy, sometimes molecules must be moved against the concentration gradient, or almost against the flow. For that, we have active transport, the topic of our next video.